And I want to make sure we have enough time here. We have quite a few questions from our audience members, so thank you for submitting them, and continue to do so. We will, like I said, we will get to as many as we possibly can. Back to the academia portion of this discussion, and around the regulatory concerns of academia versus industry. We have a question regarding do the academia have facility for GMP production? If academia isn't a manufacturing facility, they will not spend the money for GMP. And so I think that's more of a comment versus a question, but that's fine. Either one of you may take that to start us off, because this person does make a really good point.
I can definitely just get started there and turn it over. I mean, there are some well-known academic institutions that have GMP facilities. They're typically best suited for phase 1 sort of situations, and they tend to be on the maybe, I don't want to say lower end, but maybe not necessarily having all the technology. So, for example, you're probably more likely to find an academic institution that has a capability to run if you're running an adherent cell line, to run your cell stack based process, but may not have your fixed bed bioreactor type capability or something if you're looking to go that route. But yes, there are academic institutions that have a few, not all of them, but have some GMP capabilities.
Okay, great. Thank you. Curran, I want to give you the opportunity to respond to that as well.
Yeah, I think historically, academic institutions have either reached out to CMOs to make material as they start to expand into further clinical studies. And the initial material that they made, even though it was made with equipment that's not really what you're looking for in terms of long-term scalability. The processes were, I would say, remarkably robust, low yielding, but robust. And so, the real challenge to me isn't so much the GMP setting that they're using. It's more about how do you introduce a more commercial-minded process later, which might have a lower purity. And that's kind of a challenge when you think about it. If you have any findings in your initial tox studies, going in with a less pure product behind that, which is more commercially viable, is an extreme challenge. So that's the one thing I think we have to be more collaborative with academic institutions, is making sure the product purity they're producing is in line with what might be happening down the road as the program advances.
Sure. Okay. And Curran, I think this next question might be a best fit for you to start us off. What are the things you see happening in field of accelerated stability testing to work to optimize AAV formulations? And is that a priority?
Well, I certainly think there is a priority for us to look for alternate formulations that work better at -20 storage, for example. So it is a pretty significant burden for the cold chain to be sort of pinned down to a -80 freezer. We have almost 100 clinical sites that we work with for our ocular program and really see that. So when we're thinking about formulation development, we're looking at things like improving stability at higher temperatures. Frozen, but liquid formulation is not really, we've had some inroads there, but it's not something we're moving heavily towards, but maybe -20. And then I think there is work that's being done on formulation to actually help with the administration of the drug, meaning localizing where the vector goes once it's delivered, and perhaps enhancing the ultimate expression of the transgene, which is just a thought process, I think, for people at this time. But that may be an area that people explore more fully as people take the full life cycle development approach to programs that are evolving.
Okay, great. Thank you. Dave, I want to give you an opportunity to respond to that question too, if you'd like, regarding the stability testing and optimizing AAV formulations and whether that's a priority.
I think it is a priority. It's a competing priority, along with several of the others that we've talked about here today, for sure. Right? It's hard to, you don't want to lose sight on the finish line, so to speak, and the formulation and whatnot is everything. I mean, in terms of what companies are doing to try and improve robustness and things like that of their formulations. Yeah, there's no doubt that things like accelerated stability, stress stability, force degradation, and things like that. Those are tools that are, pretty commonly used. They're also, to a large degree, regulatory expectations. I know force degradation in particular is, especially in the virus world, something that is oftentimes called out by regulators and whatnot, as something that may be a gap within a company's CMC portfolio, if you will. But yeah, I mean, the emphasis, it often gets lost, I think, again, because, people think of formulation as being so robust, I think, in the monoclonal world, for example. But, a lot of work can be spent there, I think, in the virus world to enhance the overall versatility of the vector.
Good. Okay. Another question, someone is asking about the analytical testing systems. And since, Curran, you mentioned that in your previous response to the question that was just asked, I thought I'd pose this one to you first. What are the analytical testing systems used of formulation optimization?
I think in terms of formulation optimization, we look for, we do have, just maybe circling back a little bit too, it is important to know the modes of degradation of your product. And I'll give an example for AAV8 and to some extent AAV9. One of the main modes of degradation is deamidation, which also causes significant drops in potency. So when we're doing formulation optimization, we would try to pinpoint assays that are looking at deamidation on the surface of the capsid as an indicator of stability. So I think that's one example. We certainly look at more common physical attributes, given that these are 23 nanometer particles, like aggregation and colloidal stability are key things that we're thinking about when we develop formulations.
Okay, great. Thank you. Dave, I think we have time for probably one more question, and I would like to pose this one to you first, and Curran, feel free to jump in. So this person is talking about, the current role of a CDMO. And we talked about this earlier today, the importance of that partnership and what it looks like and the why behind why a developer company might partner with a CDMO. And so how do you see the current role of CDMO in current situations? What kind of baseline platforms you, as in your role, what would you expect ideally from your CDMO from a viral vector perspective?
Yeah, it's a great question. I mean, I hate to use it depends, but certainly there is a dependence on where you are, as far as I'm concerned, in your life cycle and what you're looking to get out of your CDMO. If you are a truly emerging company with a gene of interest sequence and not much else, then clearly your needs won't be quite as ambitious, let's say, as a company looking to transfer an existing phase 1 product maybe into a what you hope is your long-term, ultimately commercial provider of your material. If you're the latter of those two scenarios, then you're going to be looking for a dramatically enhanced portfolio or body of work in general. You're going to be looking, hopefully, your CDMO maybe has some regulatory experience of their own, maybe has gone through a couple inspections and whatnot, has a robust set of quality systems in place, and up to and including the equipment they're using in the GMP setting. You know, if and you want to have, you know, very in-depth technical conversations with your CDMO. How much vector are they capable? Do they think they're capable of scaling up your program too? And it's not always about scale, right? You may not need a 2,000 liter single-use bioreactor for your AAV process. You might be able to you may be able to get by with a 500 liter scale in some sort of perfusion mode or something that produces enough vector for you on a per batch basis. So I think really having those in identifying the capabilities that you need, trying to narrow down the list of the CDMOs that match, or at least you think match those capabilities, and then engaging in very technical, very in-depth conversations during the whole RFP request proposal process and whatnot, I think are, you know, critical elements.
Yeah, sure. And starting early, of course, like you said, in the RFP process. Curran, before we wrap up, I want to be able to provide you the opportunity to speak on the same topic. If a company listening was to work with the CDMO, what kind of baseline platforms should they expect?
Well, I think now for viral vectors, we're looking at CMOs typically that can operate at 200 or 500 liter scale. And I think very importantly, that they have a 2 liter model that they know can directly represent the results they'll get at 200 or 500. So when you're transferring a process to them, they can demonstrate at fairly small scale that they understand the process, the product characterization looks the same. And so I think that's really important, is to be able to minimize the scale at which you're doing that type of tech transfer checking. The other thing I would say don't miss is ultimately whether you have a good experience with the CMO or not is going to happen at the operational level. And beyond just the equipment, look at the supervisory level that's at the CMO and try to understand the level of experience they bring to bear. Because I think that's where the differentiator is, especially if you're a small company. You need to be working with experienced people. And I know they're really, really hard to find. But if you find them, hang on to them tight, because they will make the difference on your success rates for sure.
Wonderful. Okay. I do think we have time to squeeze in one more question from our audience. And so, Curran, since you've just had the floor, perhaps we could start with you. Can you let us know your thoughts on how packaging and producer cell lines are going to drive scalability?
Well, I think they can help a lot. There's great work going on around consolidation of, so we use a triple transfection. There are cell lines now that already have stably integrated plasmids, probably N of 2 and not N of 3. We don't have a fully stable cell line in place that we've been able to have success with. But being able to remove the transfection process greatly will enhance scalability of anything beyond 500 liters. And I think that will be a big step. Now, you do have to weigh making a custom cell line for every program is very expensive and takes a lot of people and time to do. So if that's what's required for a stable cell line, that's what you're buying into. We get a lot of advantage over having the transfection process because ultimately we're swapping out one plasmid. So I'm actually kind of happy where we are, but it does present a scalability question.
Yeah, sure. Dave, I want to, of course, be able to give you the opportunity to weigh in on the same thing, which is giving us your thoughts on how packaging and producer cell lines are going to drive scalability.
Well, yeah, I mean, they definitely allow, I think, for more, they open up the options. I mean, transfection in general with respect to AAV is a, famously difficult process to try and maximize the per cell productivity as far as I'm concerned. And with everyone trying to go to suspension and whatnot, I think just spatially, that just becomes a more difficult challenge when you're talking about getting small complexes to surfaces of small viruses and whatnot, and completing that entire transfection / assemblage event is just, again, very low efficiency. producer cell lines, if they're true producer cell line. I mean, there are, that term is used a lot of times. I think of a truly producer cell line as something that upon induction, like a CHO cell line, begins producing your completely assembled virus. There are some out there, I think, or at least ones that I've heard of, but more often than not, producer cell lines may still require co-infection, say, with an adenovirus or something, if we're talking about AAV, or what I call partial producer cell lines, or packaging cell lines that still require transfection with a transgene or something like that. That's not a bad way to go about things. I do think it improves efficiency. Getting one plasmid into the cell versus getting 3, I think just, the numbers say that probably should be helpful. But there's no doubt that producer cell lines in general, holy grail notwithstanding, I think in some cases definitely represent kind of the next stage.
Excellent. Okay, thank you. All right, well, that concludes this Cell and Gene Live. Thank you to our experts for the insight they've shared today, REGENXBIO's Curran Simpson and Candel Therapeutics' Dave Maheu, and as well as to our partner, Thermo Fisher Scientific, and most importantly, to all of you for your time and attention today on the Cell and Gene Live. And we'll see you next time.